Hello everybody, my name's Tom and I'm going to be the one talking with you today about some of the new innovations of our fluorometer, our 6800 fluorometer. So what we're going to cover today, we're going to cover very briefly, and you'll see how briefly here in a moment, some fluorescence theory. We're going to talk about some methodology, but we're only going to talk about the methodology in the context of how it introduces or emphasizes one of the new aspects of the 6800 fluorometer. And then lastly, we're going to have a demonstration about how to set up a chlorophyll fluorescence measurement. So before I start, I also want to say one other thing about the culture here at LI-COR. We have a tremendous amount of respect for our fellow colleagues that make instruments all over the world. We realize that we are standing on the shoulders of hundreds, if not thousands, of scientists' work who have helped to develop electronics and LEDs and all of this stuff that we take advantage of in the modern era to measure photosynthesis. We are just acutely aware of our dependence upon a lot of work that has been done in the past. So we just want to sort of acknowledge that up front. All right, so leaf level photosynthesis in generally is the exchange of gases with a leaf, shown here in this case carbon dioxide and oxygen. Light drives the uptake of CO2 and the concomitant release of the oxygen that you and I are breathing. It turns out that it's been discovered quite some time ago that carbon dioxide absorbs infrared light, and we take advantage of that.
We actually have a question, Tom.
Oh, 1 second, okay.
Somebody has asked, can oxygen also be measured by the system?
Oh, that's a good question, yeah. So while oxygen is evolved during photosynthesis, the 6800 does not measure that gas directly, no. So that's a great question. But what we do use is infrared light to measure carbon dioxide concentrations, and we infer things about photosynthesis as well as other photosynthetic parameters. But no, oxygen is not directly measured. However, it is generated during photosynthesis, and that's important for you and I, actually. The other thing is that light that's absorbed by the leaf also gives rise to a certain low amount of fluorescence off the leaf. And it's a very low amount of signal, and yet it's there nonetheless, and it's able to provide information about the what I call the electronic circuitry of photosynthesis, or what's analogous to the physiological analog of the electronics of photosynthesis. Shown here is a cross-section of leaf-level photosynthesis, and I don't really want to go into the details. I just want to remind you that photosynthesis is complex. Leaf-level photosynthesis is very complex. But what I've shown here in this slide doesn't even capture the reality. The reality is that it's very much more complex. And in fact, most of the reactions that you and I and most of the people that use instruments like the 6800 are interested in measuring processes that we really can't even see with our eye. And on a personal note, I've always found that fascinating to be able to use instruments that allow one to measure in a living leaf invisible reactions that the physical world is dependent upon. And so for me, I've found it very fascinating. So this is a picture of the 6800 system, and I say system because while we're going to be talking primarily about the fluorometer today, the reality is that the system is able to measure gas exchange also. So we refer to it as the photosynthesis system, but as mentioned, we're primarily going to talk about the fluorometer and some of the new aspects of the fluorometer in comparison to the 6440 fluorometer, the predecessor.
Hey, Tom, we actually have another question from the audience. They're asking, can all environmental parameters be controlled by the fluorometer?
That's a good question. So very similar to the 6400, the 6800 is capable of controlling all of the environmental variables. However, the 6800 does it better. Instead of, if you're like me and you wanted to do very detailed nitty-gritty research for controlling water, I always, with the 6400, would have to tweak manually the desiccant knob in order to keep VPD or relative humidity, whatever you want to measure, in order to keep that constant, for example, during a light curve. With the 6800, we've automated that. And it's one of my favorite features, actually, of the instrument is every single environmental variable is automated and very precisely controlled. So yes, it does control environment and does so much better than the 6400 does. So shown here is what's called a Jablonski diagram, and it's showing some complex reactions that can happen when a pigment absorbs light. That's the theory section. We're not going to talk any more about that. However, I do want to say something a little bit more about fluorescence because the absorption of light is a very profound thing, and I think sometimes we're so used to measuring it that we don't really appreciate what's really going on. And so on the way to work today, I had my son look up how far the Earth is from the sun. And for those of you who don't know, the Earth to the sun distance is 150 billion, that's with a B, 150 billion meters. So one can then ask, based on the speed of light, which is 3 times 10 to the 8 meters per second, you can ask, well, how long does it take for a photon to get from the sun to a corn leaf, for example, here in Nebraska? And it turns out approximately, again, these are approximate numbers, it turns out it takes about 1.5 hours to get here to the Earth. So we're talking about very grand things, photons starting at the sun and coming to the Earth very quickly, and yet still it takes a little bit of 1.5 hours. And what happens when that photon hits a pigment inside a corn leaf is something very profound. And to illustrate that, I'm showing a cartoon. It really is a cartoon schematic of an atom here. And so in the middle is the nucleus, and it's being orbited by, in this case, a single electron. And when that photon arrives from the sun, it can interact with that electron and energize it such that that electron goes to an outer or higher energy orbital, we'll call it. And that the energy in that electron can subsequently be moved on and used, in a nutshell, can be used to make chemical energy that's used by enzymes to assimilate carbon dioxide from the atmosphere into sugars, potatoes, bananas, tortillas, rice, everything that you and I consume around the world. This is where it starts. A photon from the sun, 150 billion meters away, taking an hour and a half to get here and be absorbed by a pigment at the atomic level. This is what's happening. Now what's fascinating is that, that's fascinating in and of itself, but what's even more fascinating is that we can actually take advantage of the fact that there's a certain probability that that electron is going to relax back down to the lower energy orbital, and it can do so by giving off a longer wavelength photon that we call fluorescence. And we take advantage of that. We, being scientists, we can take advantage of that fluorescent signal to understand all of these other mechanisms by which the energy is stored in chemical energy and used to assimilate CO2. So the point of all this is just to say that we're not trifling with something, some sort of just easy little thing that's measured. I mean, we're talking about very grand things, photons from the sun and atomic level phenomena. So while we're sort of, not really going into a lot of detail today, I just want to impress upon you the profound nature of being able to measure fluorescence. All right, so as I mentioned, in order to measure fluorescence, there has to be a light source. And what I've shown here is an LED output spectrum for the LEDs that are used in the 6800, and we have LEDs in the blue region at that peak at 450 nanometers, and then we have some red LEDs that have a peak at 432 nanometers. So what I'm showing here is just a simple schematic of an absorption spectrum as a function of wavelength for chlorophylls A and B, and I just wanted to point out that the LEDs that we use in the 6800, very similar to the 6400 actually, overlap in what's referred to as the Sorey, or the blue region, or at the red edge of the QI transition, or the where the red LEDs overlap. And so the bottom line is that the 6800 comes equipped with LEDs that excite in regions of the chlorophyll absorption spectrum where absorption is strong. What I'm showing here is a fluorescence emission spectrum as a function of wavelength. So this is no longer an absorption spectrum, it's fluorescence emission. And the two predominant proteins in in plants are referred to as photosystem 2 and 1, and their emission spectrum is shown here. The detection system in the 6800 has been designed to detect fluorescence predominantly at wavelengths greater than 715 nanometers. And so now what I want to do is sort of walk through putting all this together into how the measurement takes place and point out a couple of unique features about the 6800 along the way. So in the instrument is a modulated or a blinking LED, and that's used to measure modulated fluorescence. That modulated LED can pass through a filter, and when it does so, it's cut off a little bit to prevent some scattered light, but some of that scattered light is able to reach the detector. And so in front of the detector are two what are called long pass filters, and those long pass filters prevent that scattered light from hitting the detector. Now, the modulated light also elicits modulated fluorescence, and these filters are transparent to that to the modulated fluorescence, and therefore the detector is able to detect what's called pulse amplitude modulated fluorescence. Now, the continuous light, we also have some continuous red LEDs that are in the instrument, as I've already pointed out, and those light sources are able to hit the leaf. It can be scattered, and that scattered light is prevented from hitting the detector from using these filters. However, the continuous fluorescence elicited by that continuous light is able to pass through the filters and be detected as what we refer to as a continuous fluorescent signal. And so in contrast to the 6400 fluorometer, the 6800 fluorometer measures both a modulated and a continuous fluorescent signal. And so, for example, what I'm showing here is modulated and continuous fluorescent signals on the y-axis as a function of time during an induction flash. And this experiment was done by applying flashes in between 1,000 and 16,000 micromoles per meter squared per second. And so the other thing I want to point out about the 6800 is that it can deliver flashes upwards of 16,000 micromoles per meter squared per second, which in many cases is probably double what the 6440 fluorometer is capable of doing. And what you can see is that at each one of these flash intensities, there is a pair of signals. One of them is a modulated and one of them is a continuous signal. And that's probably most evident here at the highest flash intensity. You can see that there's a noisy signal, and that's the modulated signal. And then there's also a DC signal that's embedded in there that you may or may not be able to see. But just know that there are pairs of signals at each one of these flash intensities. And that's because the instrument is measuring both modulated and continuous fluorescence. And up here what I did is I quantified the dark adapted FV over FM values based on the continuous and the modulated fluorescent signals. And what you can see is that they both give values very close to what's theoretically predicted based on the quantum yield of photosynthesis. And that is that they both give a value of about 0.8. Now, many of you are probably interested in what's going on inside of a leaf under illuminated conditions. And one of the most frequently used parameters is referred to as FM prime, and it's obtained during a saturation flash. And FM prime is used to derive an assortment of parameters. I'm only going to point out a couple of them. Light capture regulation in terms of MPQ and QE, the quantum yield of PS2 electron transfer, from which estimates of electron transfer can be derived. And based on estimates of electron transfer, information about how CO2 gets from the intercellular air spaces to the site of carboxylation or mesophyll conductance can be estimated based on fluorescence-derived values of electron transfer, as well as the electron requirement of photosynthesis by comparing electron transfer with gross CO2 assimilation. And so I'm just trying to point out that this parameter is very important. This FM prime maximum fluorescence yield under light-adapted conditions is very important for a variety of different photosynthetic processes. One of the problems, however, with FM prime is that it's prone to underestimation. And so one of the things that the 6800 is capable of doing is that it has saturating flashes that are capable of going up to 16,000 micromoles per meter square per second, as I've already mentioned. And so, and that means that it's more likely possible to obtain this value. However, there's really no need for those high intensity flashes in some cases because the 6800 also comes equipped with what's referred to as multi-phase flash methodology, or MPF. And you can read about that in this paper shown down here. And I'm not going to go into any of the details. You can read about them. I simply want to emphasize that the 6800 is equipped with a flash technique that uses subsaturating flashes or moderately intense flashes in order to accurately measure FM prime. The next thing I want to point out about the 6800 has to do with the light uniformity of the light source. And so on the left is a uniformity scan of the 6800 light source, and on the right is the uniformity scan of the 6400. And what you can see, number one, is that the area is 3 times larger, 6 square centimeters for the 6800 in comparison to 2 square centimeters for the LI-6400 fluorometer. Now, in terms of quantifying the uniformity, we use sort of kind of a weird way of saying it, but we quantify it by saying that the uniformity is plus or minus 10% of the mean over 90% of the area, and that's the 6800. So for comparison, the 6400 is plus or minus 10% of the mean over 10% of the area. So as you can, hopefully as you can see for yourself, that the 6800 has been, the light source has been made much more uniform. And so with all of these new additions to the 6800 fluorometer, our aim was so that you can measure accurately and over a wide range of conditions. For example, I'm showing here a light curve for a variety of different fluorescence parameters, quantum yield of MPQ, 5PS2, just to name a couple of them. Bottom line is that we've just spent a lot of time ensuring that this instrument is capable of providing accurate measurements of fluorescence parameters. And with that, we're going to, if there are any other questions real quickly that somebody wants to send in, we're getting ready to do a demonstration, but if there are any questions.
Yeah, somebody asked,
there is a question.
What are some simple experiments to focus on if I'm just beginning to learn about fluorescence?
Yeah, it's a good question, and some simple experiments are doing just what I'm showing here. Do a light curve of all of these different parameters. All of these different parameters that I'm showing here are automated in your are automated by the instrument and are stored in an Excel file, and you can do a light curve of all of these different parameters. So, and it gives you a lot of rich information about what's going on as a function of light intensity, just as an example. Okay. Oh yeah, so just to end with, sorry about that, I forgot about one more slide here, it looks like. I did want to end with just reminding you that we've really emphasized the fluorometer today, but in reality it's a system, a photosynthesis system, and so the fluorometer is capable of not only providing information about fluorescence, but it also provides information about gas exchange. And so, for example, you can measure things like mesophyll conductance, which requires both techniques. The other thing that can be measured is the electron requirement, which is a comparison of electron transfer and gross CO2 assimilation, and the slope of that being the electron requirement. You can measure purely fluorescence-derived parameters and study how light capture is regulated as indicated here. And lastly, light capture regulation has become sort of a hot topic right now. A paper just came out recently showing that speeding up the rate at which MPQ or light capture regulation happens has been shown to increase crop yield. And so with the 6400, you can measure all the gas exchange and fluorescence parameters needed to assess things like that. So with that, we're going to move on to a demonstration.
Actually, we have one more question from the audience.
Yeah.
What is the advantage to monitor both continuous and modulated fluorescence?
That's a great question, and it's a somewhat of an involved question. So what I'm going to do is I'm going to limit my answer to saying that the modulated signal, as you saw, was much noisier, whereas in that fluorescence induction curve, whereas the continuous signal is a much larger fluorescent signal, and so the signal and noise is much better. And so if you're doing induction, if you're studying in fluorescence induction, you can get and you know how to derive the parameters from an induction curve. If you know this field, you can get much cleaner data using the continuous signal. So that's one advantage, but beyond that requires a little bit more of an explanation that goes beyond being able to do it over the air. So go ahead and email me for those of you who are interested in discussing that in more detail.
Great. Okay. I think that's all the questions we have out right now, so let's go ahead and switch it over to the demonstration.
Okay, thanks everybody. It was nice chatting with you.
So now what we're going to do is we're going to take a look at some live measurements and do some induction flashes. And so I just want to point out along the bottom of the screen here are various functionalities of the fluorometer. And for example, you'll see here that it says measuring, and that has to do with the modulation light, and it has various settings for the frequencies of the modulation that occur during the different states of the experiment. And then to the right of that are the different types of flashes that can be given. And we're going to focus primarily right now on an induction flash. And so I'm going to press that tab. And what you'll see here in the left-hand corner is various settings. And the top one has to do with how intense the flash is. And as you can see, I have it set to 16,000 micromoles per meter square per second. And for those of you who are familiar with the 6400, you'll notice right away that this is a much higher flash intensity than the 6400 is capable of doing. And also remember that it's over a much larger area, so 6 square centimeters for the 6800 versus 2 square centimeters for the 6400. So we've invested a lot of time into making the 6800 fluorometer much more improved in terms of the flash intensity and the area over which that flash can be delivered. And you also see here that the duration of the flash is 500 milliseconds. And the other thing I want to point out before we actually do the flash is I want to let you see the actual data. And so what I'm showing here is a strip chart of the assimilation as a function of time. And so this is live data coming off, and then it's moving over to the left-hand side. And you'll notice right away that this has negative values. So assimilation is showing, it's fluctuating, but it's showing about negative 1.3. And that's because the light is not on. And so this is a dark adapted leaf. And so therefore the assimilation value is not assimilation, actually, it's respiration. So the other thing I wanted to point out is the fluorescent signal. And you'll see that it also is in a nice steady state. So we've had this leaf off to my right adapting for probably an hour now. So the leaf and the instrument as a system are in a nice steady state. Okay, so I'm going to go back to the fluorometer functionality, and let's go ahead and give an induction flash. And we're going to do this manually. And there's a button here called Flash, which allows you to do manual measurements. But just be aware that you could also set up an auto program. You can see that here. You can set up an auto program that would allow you to also set up some sort of experimental protocol and do these automatically. We're going to go ahead and do it manually today. So I'm going to give a flash, and you'll see that it says in progress while it gives the flash. And at the end of that, what's nice is it shows that the flash has a unique timestamp and name. And so you can go back in the stored data and pull that Excel file out and actually look at the signal with some other software that you use to download the data with. So, but on board, you can actually look at the flash as well as the fluorescence induction. And so we're going to go over here to the very far right where it says results. And we're going to press view in order to see the flash and the fluorescence induction. So one thing I did want to note, somewhat of a technical thing, is that we designed the fluorometer to where the modulation frequency during the induction flash is 250 kilohertz. And for those of you who are slow like me and do the math, that means that a data point is being acquired every 4 microseconds. And that's important because during the induction flash, very fast things are happening. And so we've designed the modulated fluorometer to be capable of measuring those fast phenomena that are occurring. Now, as you can see here, that the fluorescence induction curve shows the typical OJIP. That's about a 40-year-old expression for these different transitions that happen during an induction flash. And what you can see is that during the first 2 milliseconds, the data is somewhat more noisy, and that's because we're doing no filtering, no averaging. And so that's a data point every 4 microseconds in order to capture this fast phenomenon. So, and the other thing I wanted to point out is that you can see that the flash intensity is also captured here in blue. And so all of that data is capable of being viewed on board the instrument, as well as being downloaded later in an Excel format. And then I wanted to go back and actually show that you can view other things about the flash. There's some quantitative numbers here. So for example, the value of FV over FM was 0.779. And for those of you who know the field, that's very close to the value of 0.8. I'll be transparent that this is not an overnight adapted leaf. It's only been dark adapted for about an hour after it sat in the greenhouse and was exposed to some other light intensity. So this technically isn't a pre-dawn measurement, but 0.779 is pretty close to what's expected from a well dark adapted plant. And so based on this, you can get both quantitative value right on board, or you can actually see the raw fluorescence induction traces. And this is all associated with the new 6800. So we're going to move on and do some light adapted measurements, and we're going to move to that section. We're going to do some light adapted measurements now. And so there are a couple of things I want to point out along the bottom of the screen here. We just did an induction flash, but the instrument is also capable of doing what's called a multi-phase flash, as well as what's referred to as a rectangular or a traditional flash. And for each one of those types of flashes, there are different settings. But before we give the flash, I want to point out what's going on. So even though we changed the light intensity that's incident on the leaf, you can see here that the leaf is in a nice new steady state, and that's illustrated by the nice constant fluorescent signal. So it's from this state that we're going to do some flashes, just to illustrate the functionality of that. So the first one we're going to do is a rectangular flash, and in this case I've set the maximum intensity to 8,000, and that's in units of micromoles per meter square per second, and the duration is 1 second or 1,000 milliseconds. And what we're going to do is we're going to do a manual flash by pushing this flash now button, But just know that you can set up an auto program very similar to the 6400, and it will automatically do those flashes. But we're going to go ahead and do this manually for now. And so I'm going to go ahead and give that flash. And you'll see that it's in progress. It's showing that the recording is in progress. And it stores that flash in an Excel file under a specific name. And so you can go back and retrieve that data. But I think it's kind of fun to go look at what happened with the fluorescence. And what you can see here, again, this is real-time data coming off the instrument. And you can see right here about 400 milliseconds ago is when I gave that flash. And you can see that there was a rise in the fluorescence. But importantly, it came back to that steady state value pretty quickly, which is a nice feature of being able to visualize this steady state, because you don't want to give these flashes too frequently. So the next thing that we're going to illustrate is the multi-phase flash function. And in this case, I've set the maximum intensity also to 8,000, and I've set some of the other parameters to very commonly used ones reported in the literature. Now, the other thing we're going to do during the multi-phase flash is we're going to simultaneously or semi-simultaneously do a dark pulse. And that's shown here. These are the schematics for that. What it's showing is that the actinic light is going to go off and the far red light is going to come on. And the length of that time in the dark is 5 seconds, and the intensity of the far red light is 25 micromoles per meter squared per second. And this allows estimation of the parameter called F naught prime, which is very important for estimating various photosynthetic parameters. And so in order to do these semi-simultaneously, I want to show you where that is done on the instrument. And so from the log files, logging options, you can choose whether or not to do a flash without the dark pulse or with the dark pulse. And so we're going to go ahead and choose that. And you can also see here that you can choose what type of flash you want to use. And I've checked multi-phase flash. And so what that means is when we hit the log button up here, up on the right-hand side, what the instrument is going to do is it's going to do a multi-phase flash as described here. And then right after that multi-phase flash, it's going to do a dark pulse. And what I think is really cool about this is that you can go over here to the fluorescence strip chart and we can actually see what happened. So let's go ahead, actually let's just do it from here. Let's go ahead and just watch this. I'm going to log a point. Give it a couple seconds. 5 seconds is that dark pulse. Okay, so the instrument, you heard those noises, and that means that the instrument gave the multi-phase flash and the dark pulse. And so what you see here is that the fluorescence rose to a maximum, and then right after that, the light went off, and you can see that the fluorescence actually decreased relative to that steady state value. And then it's very slowly coming back to that steady state value. And so let's go back over here to the results section, and we can actually view both of those. So let's start with the multi-phase flash. And what you can see here is the changes in light intensity are shown in blue, and you can see very steady values of light intensity interrupted by a linear decrease in the intensity, which is what's referred to as a multi-phase flash. And then in the red is the fluorescence. And you can see that during that perturbation of the steady state light, the fluorescence actually decreased a little bit. And that's a telltale sign that the maximum fluorescence yield wasn't actually achieved during that 8,000 micromoles during the first phase of the multi-phase flash. And it's that phase 2 fluorescence yield that allows you to estimate more accurately estimate FM prime. The other thing that we can view is the dark pulse. And so what's shown here is the actinic light going off and staying off. And you can see here that the fluorescence goes from a steady state down to the short dark adapted state in order to estimate FM prime. And so with that illustrates the light adapted functionality of the flashes for the 6800.